All right, so reviewing where we've been. So here when we have a nucleus that is filled with DNA, we would call that interphase, right? So it doesn't look like there's individual chromosomes. When we do start to see individual chromosomes, we refer to that as prophase. But here in prophase, we still are within a nucleus. Here we can also see the spindles are starting to move apart. Prometaphase, we have lost the nucleus and the DNA is moving closer towards the metaphase plate. When we have all of that DNA lined up in a single row, that is defined as metaphase. As we see that those sister chromatids being pulled apart into sister chromosomes or daughter chromosomes, um, we call that anaphase. And then post that, genetically, we have telophase, where the nucleus is starting to reform around um, the DNA, and cytokinesis, where we start getting a cleavage furrow that will ultimately separate the cell into two. So let's take a look at some micrographs. So this is um, micrographs of two cells that are undergoing this process. So this is this time-lapse kind of picture. So we have a progression of time happening here. So here we can see the microtubules in yellow, the DNA in blue, and here we can see that the DNA is condensed, but still within a nucleus. So we would call this prophase. Here, same thing. We still kind of see a circle around it. Still going to call that prophase. Here, that circle seems to have disappeared. So with the nucleus gone, we are now into prometaphase. That DNA will continue to be pulled towards the middle. Once it lines up in the middle, we call that metaphase, and this is the metaphase plate. Once we start pulling that DNA apart, we call that anaphase. And then as we start to see the cleavage furrow forming and the reformation of the nucleus, we'd call that telophase and cytokinesis. So here, let's watch this in motion. You can see that it takes a little while to kind of wrangle all of these chromosomes into that metaphase plate. So we're still in prometaphase until that happens. And once that happens, however, it very quickly progresses past that into anaphase and then telophase and cytokinesis. Also notice here that at the end of this, the DNA is starting to decondense. So we can see now how it's all not individual chromosomes. All right, real quick, closer look at this mitotic spindle. So we have the centrosome, um, two of them at either end, all those flower-like arrangement of microtubules extending out. We refer to that as the aster. We have two major types of microtubules coming off of here. So we have non-kinetic 4 microtubules that overlap each other and are actually pulling these two spindles apart. We have the kinetophore microtubules, which are actually attaching to the chromosomes. And you can see here that all of the DNA is kind of lined up into this imaginary plane, and we'd call that the metaphase plate. Now, in terms of when we get to anaphase and start talking about how we break this apart, what is actually happening is you have the microtubule attached to the kinetochore, which is the attachment point to the chromosome, you have these motor proteins that are essentially walking in this direction. And at the same time that it's walking, it is also degrading the microtubule itself. So coming back to here, we have motor proteins pushing and pulling. These are pulled into this plate through a combination of these microtubules growing and shrinking until it reaches the right point. All right, so the non-kinetochore microtubules are essentially overlapping and pushing against each other, elongating the cell. In telophase, that genetic material is being repackaged into nuclei. The cytokinesis aspect of it is when we start seeing the cleavage furrow begin and start to separate. This can actually start in anaphase. We can start seeing the beginnings of that, but it can also overlap with telophase. So cytokinesis, this forming of this cleavage furrow here, again, this is achieved through a ring of microfilaments that is contracted against in using a process very similar to our muscles contracting until we get two separate daughter cells. Now in plants, we have an added complication of the cell wall. So this process can't quite take place. Instead, what you'll see is you'll get a cluster of little vesicles forming in this interface, or in this middle in between, and it will actually start building cell wall within those vesicles. Those vesicles will continue to grow until they start hitting each other and fusing. At some point, you'll see more or less a big plate formed in the middle, and ultimately those plate comes into contact with the edges of the wall and finally separates. So here's looking at it from a plant point of view. Here we have prophase, so the DNA is condensed, it's still in a nucleus. We break down the nucleus, we're moving into the prometaphase, and the DNA is being pulled back and forth. Once it achieves metaphase, it has all the DNA lined up. Anaphase, we start pulling apart, and here in telophase, you can see this cell plate forming in the middle. In bacteria, the process is a whole lot simpler. Remember that bacteria do not have a nucleus, so we don't need to break it down. We also have a different method of packaging up the DNA, and oftentimes that DNA is a singular piece of DNA. So it's a lot simpler process, and we refer to it as binary fission. This all starts with usually a single origin of replication. So there is one spot on the chromosome where you start to see it dividing, and that continues on until we divide it in two. So we have DNA here, a single bacterial chromosome, which it tends to be circular inside of here. At one point we have an origin of replication. When we start getting replication at that site, we'll see that we have now two strands. That continues to spread apart at both forks, and we'll keep on going until we have two full copies, and then we can start dividing the cell into two using a process that's very similar to cytokinesis within our own cells. Doesn't use actin filaments, but uses something fairly similar to achieve that. And now we have two daughter cells. So concept 12.2, the mitotic phase alternates with interphase in the cell cycle. So we talked about how there is interphase, which we can't really see what's happening, and the mitotic phase, where we can see a whole lot happening. Interphase is broken into first gap, where we're prepping to do this work. The S phase where we're actually replicating the DNA, G2 where we're checking that replication for errors, and then we go into mitosis. And mitosis is broken up into prophase, where we have the DNA condensed but still in the nucleus, prometaphase, where that nucleus is now gone and the DNA is being pulled back and forth. When it's all lined up, it's the metaphase. And when it's starting to get pulled apart, anaphase, and then ultimately telophase and cytokinesis, where we're reforming the nucleus and dividing the cell into two. Again, bacteria, a whole lot simpler. We have the DNA within the cell. We start a replication fork. It continues replicating until it has two separate copies, and then we ultimately will separate it into two.